So now I think you have a good understanding of protein crystallographic technique and also learned the basic steps involved in the preparation of protein crystals for X-ray diffraction analysis. I'll now discuss the mechanism of GPCR activation following ligand binding. In this context, I'd like to introduce you to the Nobel Prize winners in chemistry 2012. The Nobel Prize was awarded jointly to Robert J. Lefkowitz and Brian K. Kobilka for their studies of G protein coupled receptors. In the next couple of slides, I'll explain what are the challenges they faced and how they resolved those challenges and came up with a very detailed mechanistic explanation of GPCR activation using beta-2 adrenergic receptors. Now, in this slide, I'll discuss the approaches to characterize beta-2 adrenergic receptor structure. The left panel is showing the amino acid sequence of beta-2 adrenergic receptor, and the blue highlighted regions are the stretches of hydrophobic amino acids that provide a clue to the membrane nature of the protein, membrane localization of the protein. Now, the biological membrane is a lipid bilayer which is highly hydrophobic. Therefore, a protein must have stretches of hydrophobic amino acids for it to be incorporated in the biological membrane. And because of the presence of 7 such hydrophobic amino acid containing domain, the proteins are called 7 transmembrane domain. Now, this figure is showing the organization of these protein domains, hydrophobic domains. There are 7 transmembrane domain which are impregnated in the biological membrane, and the N-terminal and the C-terminal sequence of the protein are highly modified due to post-translational modification. For example, the N-terminal sequence is glycosylated at multiple sites, and the C-terminal sequence is phosphorylated at multiple sites, as well as it is palmitoylated, which is a covalent linkage of a lipid group, and that also facilitates its association with the membrane. Now, in the subsequent part of this lecture, later part of this lecture, I'll discuss about the significance of this kind of post-translational modification in the cytosolic C-terminal or extracellular N-terminal of the GPCR proteins. Now, Kobilka's group, they adopted a chimeric receptor approach to study the potential ligand binding domains and G protein binding domains in the beta-2 adrenergic receptors. Now, these chimeric receptors, they used different combination of sequence from beta-2 adrenergic receptor and alpha-2 adrenergic receptor, and that allowed them to identify domains involved in ligand binding and G protein coupling specificity. Now, this is because the beta-2 adrenergic receptor and alpha-2 adrenergic receptor are both can bind to epinephrine or adrenaline, which is a hormone and neurotransmitter that signals through G proteins, and that has some action on the adenylate cyclase. But the interesting difference between these two GPCR is that beta-2 adrenergic receptor stimulates adenylate cyclase by recruitment of GS, whereas alpha-2 adrenergic receptor inhibits adenylate cyclase by recruitment of GI. Thus, they have completely opposing effect in terms of adenylate cyclase activation. And this actually made them a very interesting character to study ligand binding, to study the identification of ligand binding domain and G protein binding domain by using different types of site-directed mutagenetic approach. So they defined the map of functional domains through a series of studies using chimeric receptors and site-directed mutants. And these studies also identified intramolecular contacts and helped them to define the arrangement of transmembrane segments in the bilipid layer. Now, I'm going to show you an animation that explains the basic structure of G protein coupled receptors. As you can see that here there is rhodopsin, opioid receptor, and adrenergic receptor. All of them are G protein coupled receptors. And this animated video was obtained from Protein Data Bank, and it will show you the structure and organization of the 7 transmembrane domain in opioid receptor, which is impregnated in the bilipid biological membrane or plasma membrane. So let's play the video.
This model represents the shared architectural traits of GPCRs. With the extracellular N-terminus, the protein chain folds to form a bundle of 7 transmembrane alpha helices, connected by 3 intracellular and 3 extracellular loops, with the C-terminus reaching inside the cell. On the extracellular side, the helices form a cavity where ligands such as endorphins, morphine, or serotonin bind. On the intracellular side, the receptor is coupled to G protein. When the receptor is activated by a ligand, the G protein splits in 2 parts, which then activate other proteins in the internal signal transduction pathways.
Now, in this slide, I'll explain some basic challenges for GPCR crystallographic study, particularly protein dynamics, challenges due to the protein dynamics. Now, previously I mentioned that diffraction from a single molecule would be too weak to be measurable in X-ray crystallography. So an ordered 3-dimensional array of molecule is necessary to magnify the X-ray diffraction signal. Now, a small protein crystal may contain billions of molecules, and if the internal structure is not coherent, then the diffraction pattern will not be of good quality or high resolution. Interestingly, GPCRs are very dynamic character. That means the receptor exists as an ensemble of conformations. Now, this ensemble of conformations is depicted in this figure, where it is showing that the transmembrane domain 6 can change its position upon ligand binding, and there are multiple, it can exist at multiple positions, and which creates a broad spectrum of conformations. Therefore, if this kind of conformationally heterogeneous GPCR is used for crystallizing a protein, then this protein will be a poor quality crystal, because each conformation will have a different orientation of the transmembrane domains, and therefore the X-ray diffraction pattern will be very poor quality. Now, this was a major challenge. This ensemble of conformations or broad spectrum of conformation was the major challenge to crystallize agonist bound receptors. In the subsequent part of this lecture, I'll explain how this challenge of conformationally heterogeneous GPCR was overcome. Now, previously I mentioned that there are challenges in crystallizing agonist-bound receptor due to the presence of a broader spectrum of structural conformations or ensembles. And these usually lead to the formation of poor quality crystal, which doesn't provide a high resolution X-ray diffraction pattern. Now, this inherent problem was overcome by using antibodies and protein engineering techniques. So here I'll explain the use of antibodies or nanobodies to stabilize agonist-bound GPCR structural conformations. Now, in this figure is showing a conventional antibody structure which consists of 2 heavy chains and 2 light chains, and these are the variable region of the heavy chains and the light chains, which usually interacts with the antigen. Interestingly, camelidae, the camels, llamas, alpaca, these animals, their antibodies consist of only 2 heavy chains, so they are much smaller compared to the conventional antibody, and this green color region is indicating the variable region of the heavy chains, which usually interact with the antigen. And this fragment antibody binding segment is also called nanobodies. So nanobodies are a novel class of proteins based on single domain antibody fragments from camelidae antibodies, and they contain the unique structural and functional properties of naturally occurring heavy chain only antibodies. Interestingly, these kind of nanobodies were used to stabilize the agonist bound receptor conformations. Now, as I mentioned before, that multiple approaches were adopted for GPCR crystallogenesis. One of these approaches was the use of antibodies, more specifically the use of the antigen binding fragment from camelid antibody, which are targeted to different structural conformation of GPCRs, and these were designated as nanobodies. The other technique was the use of protein engineering. Now, the fundamental basis of protein engineering technique was the fact that majority part of the G protein coupled receptor is surrounded by lipids or detergents in case of crystallizing medium, and allowing very limited hydrophilic surface for crystal packing contacts. It has been shown that increasing the hydrophilic surface at the cytoplasmic site of the receptor can facilitate GPCR crystallization. Therefore, amino terminal T4L or T4 lysozyme fusion proteins were used to increase the extracellular hydrophilic surface available for forming crystal lattice contacts. So in this figure, it is shown that a T4L was fused to the C terminal, but at the same time, a T4L was fused at the N terminal to increase the crystal lattice contacts. And this facilitated the formation of crystals